Hey class, in this recording we're going to focus on the antimicrobial proteins of the innate immune system. And as we look at these antimicrobial proteins, it's not that these proteins are going to kill all microbes, it's just that they inhibit microbial reproduction. And as long as those microbes aren't reproducing or are reproducing at a greatly diminished rate, they really don't provide as great of a threat to our body. It really diminishes the effect that they can have in our bodies. This innate immune system is going to rely, or this aspect of our innate immune system relies on two kinds of antimicrobial proteins, the interferons and the complement system. So let's dive into it. As we look at interferons, interferons are a kind of protein that are secreted when cells are infected by viruses. So that's a key take home here. If cells are infected by viruses, certain human cells are infected by viruses, they will release interferons. And I can't emphasize this enough. The interferons themselves don't actually help the cell that produces the interferons. Interferons are a very altruistic way for the infected cell to let the next door neighbor or the nearby cells within the same tissue type to be alerted of the fact that there's a problem. So the purpose of these interferons are to alert neighboring cells and reduce the chance of those neighboring cells from becoming infected. So ultimately, as these interferons are produced, and here we have some interferon being released and exocytized, those interferons travel next door and they bind to the interferon receptor. And once that interferon receptor is activated, there's some downstream activation. No, I don't want you to focus on all these cascades. And these downstream, once the interferon receptor is activated, the protein or the cell itself will start to produce antiviral proteins. And these antiviral proteins help to neutralize the viruses that the next door neighbor is producing and pumping next door. And as we're looking at these techniques, one of the things we're trying to find out right now in healthcare is ways to activate this interferon system to help combat viral infections. Now, these interferons will cause that alert cell to synthesize those antiviral proteins. They need to also break down the viral genes. These antiviral proteins will attack single-stranded DNA and double-stranded RNA that's present within the cytoplasm of the cell. And that if we have single-stranded DNA or double-stranded RNA, there's a very good chance that's a chunk of viral genetic information. And if that viral genetic information is destroyed, the virus can't replicate. We also find that these interferons activate natural killer cells. Natural killer cells are one of the cell types of the innate immune system that destroy human cells infected with viruses. And they also activate macrophages. Because if we are having some kind of an intracellular pathogen, we're probably going to have some dead human cells that need to get cleaned up by some macrophages. So the natural killer cell will destroy the infected cell, and then that destroyed infected cell will be cleaned up by the macrophage. And these natural killer cells, the macrophages, are a 1-2 punch to kill and clean up infected human cells before they can have a bunch of brand new viron particles released into our bodies. It's also worth emphasizing that interferons can activate natural killer cells, so those natural killer cells are going to target malignant cancerous cells, human cancer cells. And we have our complement system. The complement system is not the idea that where you say, hey, you look good, hey, you look good, hey, we all look good. It's not that kind of a compliment. When we talk about a complement system, these are proteins that work together. And as they work together, they allow for a greater effect, a synergistic effect. They really complement each other in that respect. We have 30 plus globular proteins that work together for both the innate immune system and adaptive immunity. So the complement system, we could spend probably a couple hours just talking about the complement system. We don't have time for that though, so we're going to shotgun through it right now. These complement proteins are made mostly within our livers and released into the bloodstream as an inactivated form. But once the pathogen comes in contact with these inactivated or deactivated complement proteins, those proteins are then going to be activated and they'll get to work. So as we look at the activation of our complement system, it's going to bring about 4 effects. First, we can have inflammation, we can have immune clearance, phagocytosis, and cytolysis. And these are 4 different ways that we can focus on destroying the pathogen. And there are 3 ways to activate the complement system to give us these 4 different effects. We have the classical pathway, the alternative pathway, and the lectin pathway. So let's talk about the 4 effects first, and then we'll talk about the 3 pathways. Inflammation is a classic. Complement protein 3A, when activated, will stimulate a mast cell and basophil to secrete histamine. And that is a chemical that causes vasodilation, and also is going to attract more neutrophils and macrophages. And as we have more neutrophils and macrophages arriving to the scene of infection, that's going to accelerate or speed up the rate that the pathogen is destroyed. We also have immune clearance. So we have complement protein 3A. Now here we have complement protein 3B. If complement protein 3B binds to an antigen-antibody complex, an AgAb for short, those complexes are then going to be rebound to a red blood cell, and then the red blood cells circulate through the liver and spleen, and within the liver and spleen, in particular the spleen, where we have lots of macrophages, the macrophages are going to strip off the antigen-antibody complex and leave the red blood cells unharmed. And this is how we can just remove foreign antigens from our bloodstream. So this is a way, it's not necessarily a complete pathogen, it's not a whole bacteria, it's just bits and pieces, parts of a pathogen that we're going to be removing from our bloodstream with this immune clearance technique. And this is where complement protein 3B comes into effect. We also have phagocytosis, and when we think of phagocytosis, this is going to be our classic neutrophils and macrophages that are trying to swallow the bacterium and viruses. But it's very difficult for them to. They have a very incredibly hard time phagocytizing. They, for all intents and purposes, cannot phagocytize a naked bacterium. Instead, if we have complement protein 3B bind to the surface of that bacterium, that's going to opsonize it. And if we've opsonized it, we've coated that pathogen and with proteins that allow for the neutrophils and macrophages to phagocytize that pathogen. And it's kind of like, here's a good analogy. It's like putting sprinkles on a donut. And a donut all by itself, if you've ever looked at just a powdered sugar donut, you know, I'm not really that excited about eating a powdered sugar donut. But if you cover a donut in chocolate frosting, put some rainbow sprinkles on it, all of a sudden, I'm going to be much more interested in phagocytizing that donut. And this complement protein 3B is a lot like putting frosting and sprinkles on a donut. It just makes the pathogen more enticing to the neutrophils and macrophages. We also have cytolysis, the 4th method. Cytolysis is going to depend upon depend upon complement protein 3B splitting into other complement proteins. Complement protein 5B binds to that the membrane or the cell of the pathogen, and then recruits other complement proteins to form MAC, the membrane attack complex, or a MAC. And when we make a MAC, or a membrane attack complex, so I have one pictured up here for you, there are many different complement proteins involved. This MAC is effectively punching lots of holes. Each MAC is going to correspond to a single hole being punched into the cell membrane of the bacterium, making that bacterial cell wall, cell membrane, resemble a wiffle ball so that the electrolytes leak out, water flows in, and the cell pops like a balloon. It ruptures. We have cytolysis occur. So those are the 4 effects of activating our complement system. And as we look at those, just to recap, complement protein 3A is going to be responsible primarily for the inflammatory effect, whereas complement protein 3B is going to focus on activating immune clearance, phagocytosis, and cytolysis. There we go. If you have any questions or comments about complement activation and the 4 effects of complement activation, please don't hesitate to post them on the class discussion board or to shoot me an email. We will get to the 3 methods of activation in a separate recording. I misspoke earlier in this recording. And as always, happy studies.